Let me tell you a little bit more about our presenter today. So we've got with us today Dr. Susan Pell. She's the Deputy Executive Director at the United States Botanic Garden. She oversees all science at the garden, as well as the horticulture, public programs, and operations teams. As a botanist and an educator, she spent her career at Botanic Gardens and has traveled the world collecting plants for scientific study. She's passionate about sharing the awesomeness of plants with people of all interests and ages. We're very excited to have her here today answering your questions. And with that, I will turn it over to you, Susan.
Great. Thanks so much, Grace. Happy to have you here tonight. We're very excited to have our second corpse flower blooming within just a couple of weeks of each other. So you can see this very large bloom here. It's almost 9 feet tall. It's a record setter for us, the largest one we've had bloom. And it began opening this morning at around 11 o'clock, 11:30, and started at that point heating up and really kind of stinking. It's gone through a little bit of a cooling period today, and it's back heating up again tonight. So it's starting to get a little bit smelly. And we've got a treat for you tonight in that we're going to pollinate this live on this program. But I want to talk to you a little bit about this plant before we start doing that. So this is a member of the aroid family, the Araceae, which is a family that has about 3,500, a little more than 3,500 species worldwide. And it's in the genus Amorphophallus, which has around 200 species worldwide. And this is Amorphophallus titanum, Titan's arum, or the corpse flower. And you can see it has this amazing inflorescence. And an inflorescence is really a collection of multiple flowers, all attached to, in this case, one single axis. So this is an unbranched inflorescence. It's just that one straight shot there. And it has flowers attached at the bottom of this specialized inflorescence. I'm going to show you those in a few minutes when we get started with this pollination. But I want to just direct your attention to this really big bloom here to show you it has this amazing spathe. It's a modified leaf, basically, which when it's subtending or attached to an inflorescence, it's called a bract. In this case, it has an even more specialized name because it's unique to this family. This family has an inflorescence that's called a spadix, this big spiky thing in the middle that's subtended by this spade, big leafy thing. In this case, it kind of looks like cabbage. We'll take a closer look at that in a second too. But this is an inflorescence type, the spadix with the spade, is really only found in the aroid family and really defines that family. And this is the largest one. In fact, this is the largest unbranched inflorescence in the world. These things can get over around 10 feet tall in the wild, and typically they're somewhere between 4 and 8 feet in cultivation. This one's a monster for a cultivated plant, as I said, approaching 9 feet tall today. So I'm going to invite a couple of people in to start with the pollination process, and I'm going to show you this first. This is actually a vial of pollen that we got from Huntington Botanical Gardens in California. This is some pollen that they harvested from their bloom that occurred about 5 days ago, I guess. And they overnighted that to us. We're going to use that to pollinate this bloom here today. And so I'm going to bring Steve, who's one of our gardeners. He's actually an arborist, and he takes care of the aroid collection. And he's the one who gets these beautiful, amazing blooms for us. I'm going to hand that pollen over to Steve. And then I'm going to invite Jim as well. Jim is our horticulture manager. He oversees our entire horticulture operation. And he's going to help Steve. They're going to work together to use paintbrushes to remove the pollen from that little vial and paint the flowers with it. I'm going to share a secondary camera with you all to show you from our corpse flower that bloomed last week. You'll be able to see up close the flowers here. So what Jim and Steve are doing is they're going to go ahead and they're going to cut into this space. I'm going to show you that happening as well. And they've just got, basically a box cutter here. And they're going to cut into that. And what they're going to reveal is the female flowers on the lower portion of the spadix and the male flowers on the upper portion of that spadix. And this again is the one that bloomed last week, so they're drying out a little bit here. And what you can see in this is that the flowers are really reduced to only their sexual parts. So the male flowers are reduced to just being stamens, and the female flowers are reduced to just being pistils. I'm going to come back over here and show you this cutting on the spathe of the current bloom here that Jim and Steve are doing. So they're basically going to cut a viewing panel into that spathe, and that's going to allow them to get inside there, to see the flowers, and to be able to put the pollen on the female flowers here. Now, this plant, as I said earlier, was heating up, and it's heating up now again. And what that's doing is that is volatilizing, you can see right inside there, it's volatilizing the compounds that the plant is producing. And there they are, the female flowers and the male flowers above. And in doing that, it is attracting pollinators. So the female flowers are actually receptive first to the pollen, and then the male flowers will shed their pollen later. So this plant avoids self-pollination by having its female flowers, you can see here, receptive before the plant is actually releasing its own pollen. So they have to be cross-pollinated, which is what we're going to do, kind of manually tonight here. So here's Steve cutting away that other window. They're going to have two windows here so they can access both sides of the spadix and to be able to get that pollen in there to all of those flowers. And I'll show you these flowers again up close here. So what you can see, as I said, they're reduced to just their sexual parts. So here are the female flowers. You can see that sort of lighter kind of lavender colored tip there. That is right there. That's the stigma. And then the longer stalk, which is darker purple, is the style. And the bottom sort of bulbous thing is the ovary. And it's that ovary that's going to become the fruit. So they're going to paint the pollen on the stigmas. Again, that sort of lavender colored thing. And then the pollen will grow down the style and pollinate that ovary. So here are those windows. You can see there's light coming in from the other side here from where Steve cut. And there are those female flowers. And you can see the colors are different here in these fresh flowers. That stigma is a sort of a light green color. And you can see there's the styles are sort of glistening there. They have some little hairs on them. So Steve is gonna pollinate these in just a second here. I'll tell you a little bit more about the spathe while we're waiting. So the spathe is what really allows those compounds to travel far once the plant starts heating up. So it's going to essentially sort of like a trumpet, it kind of expands the sound. It does it with the scent. So this scent will actually, once this plant is heating up fully, that smell will travel about a mile away, and that's going to be attracting pollinators. And those pollinators are going to be pollinators that are normally attracted to carrion, to rotting meat, to dead animals. And the reason this thing is called a corpse flower is because those compounds are super sticky. They're mostly sulfur compounds, and they really, really smell like rot. And also, as we learned last week, too, or we saw a smell last week, they smell a little poopy as well. So here we've got Steve. He's pollinating here. He's transferring the pollen from his vial onto the stigmas. A little bit sticky. So the pollen's a little sticky when it comes out, and maybe I can get Libby to show you the picture of the pollen releasing from the stamens, which is a really cool thing. Basically, the pollen comes out in these sort of long strings, and the pollen comes out in these long strings, and so it's really quite sticky. So when we harvest it, and we'll do a harvesting of it tomorrow, that harvesting will happen basically from a kind of a little boat that we put down underneath, a little sort of a collection basin that we put down underneath the male flowers. And you can see Libby's got a picture up there. If you look at the left side of that photo, you'll see those long strings hanging down. And those long strings are the pollen. So they're quite sticky. After we harvest it from the sort of wax paper basin that we put down below it, we will dry that out, and then we'll freeze it, and then we'll ship it to our partner gardens. All right, now I'm going to share my camera again here.
Susan, can I jump in with a quick question from our audience?
Yeah, absolutely.
Can you help us understand what's the difference between an inflorescence and a flower?
Yeah, that's a great question. So a flower is kind of the smallest complete reproductive unit of a plant. And an inflorescence is a collection of multiple flowers. You can see Steve here. So in this case, each of these little yellow dots that you see there, each of those long pink things, those are individual flowers. And then if we look up a little bit, it's a little harder to see. Let me show you on the other inflorescence here. So on this one, you can see these little yellow dots here. Each of those yellow dots is a male flower. And there are hundreds and hundreds, you can see that here, hundreds and hundreds of flowers on this inflorescence. And as Jim and Steve are pollinating here, oops, sorry about that. You can see all of those many, many flowers. Each of those stalks that you see is an individual flower. And this is a two-person job because you've got to cut 2 windows into this. And then tomorrow, when we actually remove that spade, we'll need about 3 people for that because we have to have 1 person on each side of the large spade holding it because it's quite heavy, and then 1 person cutting it off.
And Susan, did we manage to get our pollen to Huntington?
So we did get some pollen there, but we did not. The question, Jim, is did we get our pollen to Huntington?
We sent it to them, but not in time for their bloom.
We sent it to them, but it was too late for their bloom.
They are going to cryo-freeze it for their next
bloom. So they are freezing it, they're drying it, or they're freezing it rather to save for their next bloom. They actually have a large collection of corpse flowers, and so they'll be preserving it for the next time they have a bloom.
And Susan, can you help us understand why are we sharing pollen with other botanic gardens?
This is a great question. So we're sharing pollen with other gardens because this is part of our conservation program here at the U.S. Botanic Garden. What we are interested in doing is maintaining the genetic diversity of the corpse flower in cultivation. This is a plant that is rare in the wild and has been in cultivation for quite some time. It is becoming slightly more common in cultivation. And what can sometimes happen with cultivated plants is that you might just be sharing kind of offshoots or their genetic clones of each other, and then you kind of you don't end up with that much genetic diversity within the cultivated sort of population of the plants. So we've been partnering with Chicago Botanic Garden, who's doing a genetic study of corpse flowers, and we've given them samples of every one of our plants. We have about 15 mature plants in our collection, and around 50 total with a lot of seedlings from our 2017 bloom that we effectively pollinated and got seedlings from. And so we're sharing the pollen out to help maintain that genetic diversity, and we're tracking that through this effort that's led by Chicago Botanic Garden to really look at the genetic diversity of these things using some DNA sequencing technology to understand the diversity within the plants, and then to be able to identify the parentage of the offspring through successful pollination efforts.
Thank you, Susan. And we've got another related question. So is there a reason we're not using the pollen of the corpse flower that just bloomed last week to pollinate this corpse flower?
Yes, there is. And the reason for that is that these plants are actually siblings. And so we don't want to breed them with each other for obvious reasons, I guess. So we're interested again in that genetic diversity, right? And so we wanted to really get as distantly related corpse flower pollen as possible. So we shared ours out, and then we used pollen from other gardens to pollinate both the first bloom and the second bloom that we have today. And it's a little bit early for us to tell whether or not our first pollination effort was successful. We're getting a little bit of swelling happening in the ovaries here, but it's going to probably take several weeks for us to really be able to tell if that first pollination attempt was successful. And we actually used the pollen from several different plants to pollinate this one. And again, we'll sort of after the fact look at the genetics of the offspring, if we get any, to see what their parentage was. We know the sort of lineage of the parent pollen that we used in all of these cases, and so through genetic analysis, we'll be able to tell which of the offspring were from which pollen. We've got a beautiful picture here up of an almost mature, what's called an infructescence. So once an inflorescence goes into fruit, it then becomes an infructescence. And this one is, I say it's almost mature because these will turn a darker color than that. They'll be a sort of a dark orange or red color So here they are sort of yellow to light orange, not quite mature yet. And that process of maturation of the fruit takes about 6 months. So from pollination to mature fruit where we can sort of harvest them off of that stalk and start planting them is about 6 months.
And Susan, is this inflorescence, this corpse flower that we're looking at, is that at peak bloom?
That's a great question. So this one's been a little bit weird kind of the whole time. And you can see on our live cam, and you can also see here that the spadix itself, so the long stalk in the middle here, the spadix itself is really kind of folded in on itself. And it's very wrinkly at the top. So if you look at our live cam, you'll see very much that wrinkly top on the spadix. The other thing that's happening that we can see here and can see on the live cam is that the spade, although it's been opening most of today, it's sort of opening on one side, kind of the front and the right side on your view, and the left side is really staying close up against the spadix. Now, that may change. It may fully open later tonight. I would encourage you to sort of stay tuned to our live feed on our website to see what happens later. It's anyone's guess, really, if this is going to fully open or if this is what it looks like fully open. Corpse flowers, when they open, will sort of go anywhere from sort of just almost like a tulip shape to a full-on, trumpet shape, really flat. And the first one that we had bloom, this one on the right, you can see a picture of it there. That one really went fully flat. So it opened up, almost pretty much opened up all the way, and the spade really flattened out. So the top of it was about 4 feet across. And in doing so, again, really spreading those compounds that attract its pollinators. And we do see a lot of flies visiting it here in the conservatory. But some of these, as I said, they really kind of open up just barely. They barely will pull away from the spadix, and then they'll close back in. And so we would anticipate this one would open up a little bit more overnight, and then would probably be closing back up in the time after that. And you can see here is a picture of the one that we've got on display right now that's opening, and you can see on that sort of right side, it's pulling away from the spadix, the spade is, but on the left, it's really staying close up against that spadix. And it may remain like that, or it may fully open up. And sometimes, of course, they don't open at all. That's always a concern.
Thanks, Susan. And what happens to the spade and the spadix in nature post-pollination? Do they just sort of fall away?
Yes, and so we actually will cut them off here in cultivation, and we do that because we're growing them in much less humid and less hot conditions. In nature, they will quickly rot away. So basically, the spade will close back up, the spadix will fall down from the top, and then eventually that spadix and spade will rot away, and it will leave essentially what we have kind of exposed by removing the spade and removing the spadix. So I can show you the one we removed last week. This is the spadix that we removed, and it's got some cool tissue you can see inside there. Yeah, really neat plants. This one had sort of slumped over and kind of flopped over, so we removed it. And then here is this is the spade that we removed last week. It's part of the spade, I should say, a tiny part of the spade. And basically, when we remove the spade, we cut away the top portion of it. So we'll cut away this kind of top part of it and remove that. And then what we're left with is sort of a collar. This is the one from the previous bloom. We're left with this kind of collar that will then further cut off. And the reason we don't start with cutting the whole collar off is that this is the heaviest part of that spade. And if we were to just have if it would slip out of our hands or something and we still had part of this attached, it could actually pull the whole plant over, essentially pulling the corm out of the pot. And the corm, if we can show a picture of that, is the underground part of this plant. It's a storage unit, kind of like a potato or a carrot. It's an underground part. It's stem tissue, very hard stem tissue. And that structure is just underneath the soil surface. You can see there in the center of that picture, that's a rotting leaf that was cut down, and the rest of the leaf sort of rotted away. That will stay in a dormant period for a number of months, and then it will either grow a leaf or it will grow an inflorescence bud. And we never really know until it's a little bit taller, whether it's going to be an inflorescence or a leaf. Steve actually did a great video that's on our Corpse Flower website that talks about how he knew when the first one of these was an inflorescence and not a leaf. So definitely watch that to learn more about that. But either makes a leaf or an inflorescence. You can see the life cycles up there. It starts off as a corm in that dormant stage, grows a bud, either becomes a leaf or an inflorescence. If it becomes a leaf, what that leaf is doing is photosynthesizing and building up the nutrition that's stored in that corm. It's making that corm bigger and bigger. And some of our corms are, 40 pounds or more. They can really be quite large. And then when it's making an inflorescence, it's using up that stored energy. So this is really not adding much at all to the corm. It's actually sucking out the energy that's been stored up through the several cycles of producing a leaf. And how much does this one weigh, Steve? This corm about?
Probably about 60 pounds.
So this one, they said, is 60 pounds. And you can see there's a leaf stage there. It's one of our education specialists standing next to the leaf. And it looks like a tree. I realize that. It looks like it might be a full tree, but that's actually just a leaf. That big sort of trunk-like thing is just the petiole or the stalk of the leaf. And it's got lots and lots of leaflets above it. It can be about 20 feet tall, about 20 feet wide. So it's an enormous leaf, but just a single layer.
And Susan, this question, I believe, is related to the underground structure, but definitely calls for a morphologist. So does this species produce, I hope I say this correct, bulblets?
Oh, yeah. So it does have what we would call offsets or shoots that can form on the corm. And we actually have one large one at our production facility that had some damage to the apex. So that what's called an apical meristem, where that bud is going to be produced each year. It had some damage to it. And what's really interesting is that what we're seeing is tons of sprouts all around it. So it's actually produced many offsets. And so really reproducing what we would call asexually reproducing or vegetatively reproducing to make lots of clones of itself. And in fact, I believe it's the first one that we had here bloomed is actually a clone of another plant that we have, which is one of the offsets that we harvested and separated and repotted.
Thank you. And we've got a question about the leaf. Is the leaf smelly at all?
The leaf is not smelly at all. So the only purpose of the smell in these plants is to attract pollinators. So it's just to volatilize those sulfur compounds, heat it up, spread them around the forest and attract those pollinators. And I've given this analogy before, but if you can imagine, your home and you're chopping up garlic, the garlic smells really strong to you standing right there by it, but the people in the living room or their bedrooms really cannot smell that garlic. But once you put it in that hot pan, it's going to spread that garlic smell throughout your house. The same thing is happening here. When these heat up, they're volatilizing, heating up those compounds and spreading them out throughout the air. The pollinators from a mile away can smell them and be attracted to them, thinking that it's a very large dead carcass.
Thanks, Susan. And we've got a great question here. Why is this plant important?
It's a great question. So, I would argue that really all plants are important in that they, photosynthesize and produce oxygen for us. We wouldn't have the atmosphere that we have without plants. We wouldn't be alive without plants. They are the foundation of the food web, the ecosystems. All animals ultimately rely on plants. I love when I meet somebody and they say, I don't like to eat plants. And I say, well, everything you eat is really a plant. So even the beef, that you have, the burger that you have, the bun is directly made from plants, and that cow entirely eats plants to live, right? So they're just all extremely important. This one in particular, though, I think is just really charismatic. It's a rare plant, so that makes it interesting. It grows in a, kind of a mysterious place to a lot of people, Sumatra, Indonesia, and it's also just enormous. So it's the largest unbranched inflorescence in the world. That is this giant, crazy leaf, and it smells like a rotting corpse. So all of those things just make it just super cool, basically.
Thanks, Susan. Well, with that, we are just about at 7:30. So I just want to say thank you so much, Susan, for sharing your knowledge with us. And I want to say thank you so much to everyone who came to ask questions today for our live Q&A and dissection.
Awesome. Thanks so much for joining us.